Hello, it's Mr. Sang here again. In this video, I'll show you how to use the virtual microscope. When you get to this page on my note, you will click here, and that will take you to the BioNetwork virtual microscope. Now, in this virtual microscope, you can do a number of things. You can click on any of the 5 buttons on the bottom, but we're going to stick with the first 3. In the guide, you'll notice that it's a reference guide and you can refer to this whenever you need to, and it shows you what the different parts are of the microscope. If you click on overview here on the left, it starts off with an introduction and it tells you what each part is and what each part does. So here we are, and there's the microscope, and we're going to click on learn. And when you get to this part of the virtual microscope, you can see that they have all these question marks everywhere. By clicking on the different parts, you can see what they're called and again, another definition or a description of how to use it. This is the lens paper that I just told you about, and this is the immersion oil that you use for the high power lenses. This Kim wipes is the laboratory's version of tissue paper, Kleenex, and it's used for lots of things, but not for cleaning the lenses of a microscope. Down here we have our slides, and this is where we see our next step of this demonstration. So let's go back out to the main, and we go to explore. So what we're going to do here is we're going to take a look, and I'm just going to show you how to get started. Then I want you guys to take a look at different microscope slides to see what they look like. Here is the letter E. This is the view that you get. It's not actually in focus. So you take the coarse adjustment focus, the coarse focus, and you put it into focus first. That's always your first step. You notice that if you go too far, you'll be out of focus again. So this is a 40 times magnification because we're going to multiply these numbers by 10. If we move the fine focus, you might see some difference, but it's very minimal. We switch to the 10 times magnification, which equates to 100 times magnification of the letter E, and you'll see that you're much closer. The letter E is upside down, and that's an interesting note that everything you're looking at inside the microscope will be upside down because of the movement of the light through the lenses. Okay, so let's remove the slide and let's look at a plant slide. And let's look at the onion root. And again, it is out of focus, so we move the coarse focus until we're in focus, and we can move the slide to whatever we want to look at. Then we can change the magnification to the 10 times. So you notice that the objective lenses are moving when I change the magnification lenses. So at this point, what we're going to do is we're going to look at these cells and notice how that they are actually quite square as opposed to being more irregular in shape. Those little dots are the nuclei. Nucleus is 1, and then plural is nuclei. So these are all nuclei that have been stained by some sort of stain. As we increase our magnification to 400 times, we can start to see that there are differences between the different cells. And we're going to talk about why this cell here looks so funny and why some other cells seem to have just a regular circle in there. So let's add our drop of immersion oil, and that's done by clicking on these 2 bottles, by the way. And we can't really see, it's too dark. It seems kind of blurry too. Ooh, look at that. There they are. Those are chromosomes. And these chromosomes are in the process of dividing. Oh, those are beautiful samples there. Okay, so what happens if I use the coarse focus when I'm in the 100 times? Oh, I should avoid using the coarse focus knob with the 100 times objective because it will crack the lens, and I have just ruined the microscope. But this is just virtual, so you can start all over again. Okay, so I'm just going to show you one more, and this is the animal slides, and we'll just take, I guess we'll take a spider. Ooh, what does a spider look like under a microscope? So this is probably the spider cells and not the spider. It is the spider. Okay. I didn't think the spiders are that small. So this is 40 times magnification of a really tiny spider. And you can take a really good look at those 8 legs. That's why they're not insects. And as I go to the 100 times, click on the bottles to add the immersion oil. And whoa, that is crazy. At this magnification, you can almost see through the spider, and you can see how those are cells. You can see the cells of a spider. Wow. Okay, so I'll let you guys play with all the different options that you have here, and that's all for this video. Thank you very much for watching.